We up on Zoom? Okay. So, and where's my Zoom audience up there? Okay. All right, well, it's my great pleasure to introduce Justin Hockett, who is defending his master's thesis today here at Oregon State University. And there's the title there, but I've got a few slides I snuck into his slide deck here just to kind of get things going. Nothing that's going to be too embarrassing, so don't worry. Okay. That's it. So let's run the reels all the way back to 2018, which is when I first met Justin. And Justin was this sort of bright-faced kid right here, second from the right, squinting in the sunlight, in the aquatic entomology course that we teach out of my lab group. So over 5 years ago, he was an undergrad right there. And here we are collecting aquatic insects at PV Arboretum. And Justin was one of the superstar undergrads in that course. So one of the people who really got the taxonomy, dialed it in, did really well. I think he got an A. Okay, good. So if you didn't get an A, you should have gotten an A. But given that track record, we recruited him into the lab soon thereafter to just help with taxonomy on another project on the River, in which he did really, really well. And so fast forward through time, Justin became an important member of our lab group, and eventually when an opportunity arose, pulled him in to become a graduate student because it was just sort of a logical succession. So he was already familiar with the Verde River ecosystem, which you can see back here, and you're going to hear about today in his presentation. But he got better and better at it, and really ultimately became kind of the star go-to insect taxonomy person in the lab group. Like at any given time, there's one person who really knows their stuff and is the go-to person, and that's been Justin, actually for a number of years right now. But he's also been an important person in the field in terms of teaching other people field methods, just a really good soul to have along in any kind of field work. And also, I looked for it, and I swear I have it, but Justin is just a radical cliff diver. Like you wouldn't expect it because he's kind of calm and everything right here now, but this guy just loves to jump off of really tall cliffs. And so this is the Colorado River right here. I think either right before he just plunged into it from about, I don't know, a good 25 feet. Anyhow, sort of fearless in his own way. And so here we are, a little more facial hair and a few tattoos later. I have to say that Justin has just been an integral part of the lab, and he's been there for all of us in any kind of weather. And so with that, I want you to take it away.
Sounds good. Thank you for that, Dave. And I want to thank everybody for being here, both in person and virtually. It means a lot to have people here supporting me. So with that, here's my thesis title, From Hydrology to Biology, How Abiotic Factors Structure Invertebrate Communities in a Free-Flowing River Network. Here's a little roadmap for my talk. So first I'll go over some introductory material, and then I'll go over my research questions, which will include some of my results. Then I'll go over some of the implications of my research. So first, I'm going to start off with this question: why study rivers? So I think the first and most obvious reasons are that humans rely on rivers for a lot of different things, including agriculture, clean drinking water, and recreation, among a lot of different things. But freshwater ecosystems are also biodiversity hotspots. So freshwater covers less than 1% of the Earth's surface, but contains around 9.5% of the Earth's animal species. And rivers are a small percentage of that surface freshwater, so about 0.5% of all surface freshwater are rivers. So rivers are important because they contain a disproportionate amount of diversity relative to their area. And these river ecosystems are also under threat from things like dams and water withdrawal that alter the integrity of river ecosystems and also prolong drought, especially in the American West. So that brings me to our study system. the Verde River in Arizona. So you might be wondering why Dave and I have been saying Verde and not Verde as it is spelled. Well, the local people in Arizona that live around the Verde, so those communities, they call it the Verde. So in order to be authentic, we're just going to roll with Verde. So the Verde is located sort of centrally in Arizona, northeast of the town of Phoenix. And you can see on this map, we have 3 study sites on the Verde River at Beadley Flat, Childs, and Sheep Bridge. You also might notice part of this, this river is highlighted with red and yellow. So this is the stretch of river that has wild and scenic protection. So this is a legal protection that states that this section of river must be maintained in a free-flowing state. So legally, dams aren't allowed to operate above this reach. And like I said, it must be maintained in a free-flowing state. But below our last site at Sheep Bridge, there are a couple of dams, but they don't influence this section of river. The Verde is also experiencing declining flows due to things like human water use in the area, so things like agriculture and municipal needs for growing communities, and also drought, as I mentioned. So a lot of rivers in the American West, the Verde River included, are experiencing declining flows due to drought. So I want to introduce this idea that rivers are spatially complex ecosystems. So rivers contain different habitats that have different physical and chemical properties. So if we look at this diagram, I've circled a riffle, a pool, and a run. So these are 3 different habitats within river ecosystems. And these are going to harbor different species due to the adaptation of these species to these physical properties. So a major division in these habitats are between erosional and depositional habitats. So erosional habitats are going to be our riffles and runs. These are going to be fast-flowing habitats that contain larger substrates. So any fine substrates that might exist in these habitats are just going to get washed downstream, where they end up in depositional habitats. So these are going to be our pool habitats. These are generally slow-moving, and they contain these finer substrates. So we ended up classifying these habitats with discrete definitions so that between our sites, we can be sure that we're sampling in a similar area. So for erosional habitats, we define these as shallow water, less than ½ meter deep, and fast, so greater than 0.2 meters per second water velocity. Similarly, we have runs, which are deep, so greater than ½ meter deep, and fast, greater than 0.2 meters per second. And then we have our depositional habitat pools. So these are going to be slow areas of the river, so a water velocity less than 0.2 meters per second. And these are going to be associated with the stream margins. That brings me to the organism that we studied, which are aquatic invertebrates. So these are adapted to a wide variety of different in-stream conditions. And there's a lot of literature on aquatic invertebrates that shows that they're adapted to erosional and depositional areas in the stream. So for instance, we have this mayfly on the left. This organism is going to be pretty streamlined and flattened. It's going to be good at clinging on to rocks in these fast moving areas. And on the right, we have this dragonfly larva. It's going to be quite a bit larger and it's a little bit more bulky. It's going to have a hard time persisting in a riffle, for instance. So that's going to be a pool insect. And aquatic invertebrates are also diverse. They contain many different taxonomic groups. This includes insects, crustaceans, mollusks, worms, and many others, but these are sort of the main ones. But aquatic insects are important because they have a terrestrial life stage. So a juvenile mayfly is going to be aquatic. It's going to have gills. It's going to have to remain in the stream. But in order to reproduce, it's going to emerge into this terrestrial adult phase. It's going to be able to fly around and find a mate before depositing its eggs and dying. So that's how it completes its life cycle. Whereas other aquatic invertebrates are going to lack this terrestrial life stage. they essentially have to remain in the water for their entire lives. So this is important because aquatic insects are food for a variety of terrestrial consumers. So this diagram from Baxter et al. 2005 does a good job of illustrating this. So you can see in the center of the screen, we have our aquatic insects. They're going to be food for things like fish. At some point in their life cycle, they're going to need to emerge from the stream, and then they're going to be eaten by a variety of things that live near the stream, including lizards, birds, bats, spiders, really anything else that wants to eat insects. And studies on the Verde have proven this empirically. So this study from Baruch et al. 2021 showed that aquatic insects are an important food resource for terrestrial consumers that live near the Verde River. So we can see in these graphs, so if we just look at that lime green and that orange colored part of the bar graph, that is the proportion of the diet of this wolf spider and this ornate tree lizard that is made up of aquatic insects. We can also see from this graph that these aquatic insects are important for cycling nutrients into the terrestrial ecosystem. So these larval insects are eating things like algae in the stream. And they're going to hatch out, and those nutrients from the algae are going to be basically when things eat the invertebrates, they're going to get those nutrients. Also, leaf litter from the terrestrial ecosystem is going to fall into the stream. And similarly, these aquatic insects are going to eat the leaf litter and recycle it back into the terrestrial ecosystem. So that brings me to my research questions. So question one is, how is aquatic invertebrate diversity partitioned within the Verde River? Question two, how do macrohabitats differ in terms of diversity and abundance of aquatic invertebrates? And then lastly, question three, how will aquatic insect abundances change under reduced flows? So we're going to start with question one. How is aquatic invertebrate diversity partitioned within the system? So before we get into that, we're going to have to talk about how we sampled invertebrates. So we have Dave here demonstrating proper DNet technique in a riffle or a run. So when we take an invertebrate sample, we're really getting a snapshot of the invertebrate community in a location, both in terms of diversity, or the different types of invertebrates, and abundance, or how many of these different types of invertebrates are we finding. So when we're sampling a riffle and a run, it's pretty simple. We lodge the DNet into the riverbed, and then we scour 3 square feet in front of the DNet to dislodge these invertebrates, and the stream energy will carry them into the net. But when you sample pools, there's going to be little to no flow in these areas. So we had to devise a different technique. So instead of the stream doing the energy, we have to provide the energy. So we utilized the sweeping technique where we sampled about 6 square meters of habitat. And we sampled the stream bed, the water column, and the edge vegetation equally for about 60 seconds. We then took these invertebrates back to the lab where we identified them to genus or family level. So basically one step above species. And we found 100 different invertebrate taxa throughout all of our sampling on the Verde. So now let's talk about how we partition these samples within the river. So we have, like I said before, we have 3 sites in the Verde River, one at Beasley Flat, Childs, and Sheepridge. These sites were chosen basically due to access. Once you get downstream of Beasley, it's pretty wild country out there. There's not a lot of areas you can get down to the river. So these are basically it. Alternative would be to float the river, but we didn't do that. So I've sort of sketched out our invertebrate sampling regime here. So hopefully it makes a bit of sense. So we have 3 river reaches, 1 reach at each site. And within each reach, we have our 3 habitat types, riffles, runs, and pools. So we sampled 1 riffle, 1 run, and 1 pool in each of our reaches. And then within each habitat, we took 3 invertebrate samples. So we sampled invertebrates 3 times in each of our habitats. Then we repeated this whole thing 5 different times. So starting in fall 2017, and then alternating spring and then fall, until fall 2019, we took tons of samples. So if we do this multiplication, we get 135 different invertebrate samples. So I'm going to talk about our first statistical method for how we want to analyze this data. So I'm going to introduce the idea of a PERMANOVA, or permutational analysis of variance. Essentially, this is going to tell us if our samples grouped at these different scales are different. So this is basically going to tell us, are there differences between our macrohabitats, riffles, runs, and pools, between our study reaches, or between our sample events, or when we went down to take these samples. It'll also tell us which of these groupings explains more of the variance between our samples. So in other words, this is going to tell us which of these groupings is the most influential to invertebrate community structure. So now we're going to look at this complicated looking table of the PERMANOVA results, but I'll walk you through it. So first, let's look at macrohabitat. So we can see it's significant, has a p-value less than 0.01. But let's focus on the R-squared values. This is going to be the explained variance. So we can see that macrohabitat explains about 40% of the variance between our samples. Now let's look at sample events. So the timing of when we went down to take samples. This explained about 14% of the variance. While the study reach, or the location where we took our samples, this only explained about 6% of the variance. So really, macrohabitat is the most influential variable in structuring these communities. So now we're going to look at this graphically to just see how these samples compare to each other. So this is an NMDS ordination, or a non-metric multidimensional scaling ordination. And it's basically a way to take these complex ecological communities and sort of distill them down to an hopefully an easy to understand graph. So all these points on this graph represent an invertebrate sample that we took. And the distance between these points indicates how similar the communities are in these samples. So if samples are close together, that means we found similar invertebrates in these samples. If these points are far apart, that means the invertebrates we found are quite different. So right off the bat, we can see a clear distinction between erosional and depositional communities. So we have our riffles and runs. The erosional samples are on the left side of the ordination, and our pools, the depositional communities, are on the right side of the ordination. We can also look across that horizontal axis between our macrohabitats, while the axis 2, or the vertical distribution of our samples, is separating differences within our macrohabitats. I'm not going to get too much into this, but the vertical distribution of our points was related to the timing of our samples. Okay, so some takeaways for our first question. How is aquatic invertebrate diversity partitioned within the river? So first, we found that the macrohabitat scale is the most influential scale to these invertebrate communities. We also found that invertebrates separated into clear erosional and depositional community types. So this is something that's been shown in a lot of the literature, and it's cool that we saw such a clear result. That brings me to question 2. How do these macrohabitats differ in terms of the diversity and abundance of aquatic invertebrates? So first, we're going to look at this basic table that I made, showing the total taxonomic richness and the mean taxonomic richness in each of our habitat types. So we can think of total taxonomic richness as the total number of species we detected across all of our samples in a given habitat. While the mean taxonomic richness, we can think of this as the average number of species we detected in our different habitats. So let's take a look at the total taxonomic richness first. So we can see that pools contain overall more species than our riffles and runs. But if we look at the mean taxonomic richness, or this per sample diversity, we can see that pools have a significantly lower taxonomic richness than riffles and runs. So overall, pools contain more species, but they're more variable. And we can see this on our ordination again. So if you look at these pool samples, they're highly spread out on the ordination, indicating that our samples and pools are pretty different from each other. Whereas if we look at our ripple and run samples, they're tightly clustered, which means that these samples are pretty similar to each other. Now let's talk about indicator species analysis. So this is a way for us to tell what species are associated with ripples, runs, and pools. So indicator species analysis takes into account the fidelity, or how consistently do you find a species in a particular habitat, and the specificity, or how exclusive is a species to a particular habitat. And this will give us an indicator value between 0 and 1. 1 would indicate a perfect indicator. We always find it in a habitat, and we only find it in a habitat, where 0 would indicate a particular habitat. So let's start with the indicators of riffles. So we found a lot of indicator species, and 11 of which were strong indicators, which means they have an indicator value greater than 0.5. So those are the taxa that I've bolded here on this table. Now this probably just looks like a bunch of random words, and you might not know what they mean, but these are the different invertebrates that we found. And the important thing to know that all the indicators that we found within riffles are these terrestrially available insect taxa. So these are all the larval forms of these insects. They're going to have the indicators of runs. So within runs, we only found 10 indicators, and only 1 was a strong indicator. But 9 of the 10 were these insect taxa. So I have starred one of these taxa here. That would be the non-insect invertebrate freshwater clam. So it's kind of interesting that invasive species have strong indicators. It sort of takes us back to that variable nature in pools. So we were detecting these species in pools, just some of them we just didn't detect very often. And 19 of the 24 indicators of pools were also these insect taxa. If we look at the bolded taxa here, we can see that I've starred 2 of them. One is actually the taxa with the highest indicator value, which is a freshwater snail. We also have an amphipod crustacean as well. Insect taxa, we found that riffles harbored much higher abundances of insect taxa than our runs and pools. So looking at this graph, on the y-axis, we have the mean abundance of insect taxa. So think about this as like the total abundance of all individuals that are insects within a sample. And within riffles, we found about 3,000 on average in a given sample. Whereas runs, we found about 1,300 or so, and pools about 700. So these riffles are really acting like insect factories, like cranking out tons of insects. So here's some takeaways for question 2. How do macrohabitats differ in terms of diversity and abundance of aquatic invertebrates? Aquatic insects have a high fidelity and specificity to riffles, and that riffles harbor greater abundances of aquatic insects. But we also found that pools are important for regional biodiversity. So while we found less insects in pools, we found other things. We also found that pools were more diverse overall than riffles and runs. This brings me to my last scientific question. How will aquatic insect abundances change under reduced flows? So in order to do this, we had to do some hydraulic modeling in the Verde River. And by we, I mean Dr. Chris Holmquist Johnson, the USGS. So essentially, he went out to our field sites and he took some drone imagery. So he flew a drone over the entire reach and he took some images and used a technique called structure from motion, which essentially extracts data from these images, sort of looking at the differences between these images and how the river is moving. So this data paired with river gauge data, he was able to construct these 2D hydraulic models. And these had a 0.3 meter cell size, which means that every 0.3 meters of river, we had a datum. So with this, we were able to calculate and map macrohabitat areas directly onto our study beach. So as you can see here, I've color-coded our different habitat types on this map. So we have riffles that are orange, runs that are red, and pools that are blue. And this allowed us to make predictions of macrohabitat area under different scenarios. So let's take a look at that. So this is looking at the Beasley Flat Reach only, which is the only reach where we have all this work done. Still working on getting the other ones done, but we'll just roll with these. So if we look at that top map, that's the 4 cubic meter per second discharge. So what that means is that this map is showing what the macrohabitats will look like when 4 cubic meters per second of water are flowing down this river. And this is the baseline because this is around the discharge of when we collected our invertebrate samples. So starting here, we lowered our discharge by 1 cubic meter per second intervals until we get down to just 1 cubic meter per second. So you can see there's a dramatic loss in our riffle and run habitat, and pools are just starting to take over this map. So let's take a look at this graphically. So this is showing the exact same thing. We have our estimated macrohabitat area on the y-axis, and our x-axis is the modeled baseflow discharge, which is starting at a baseline value of 4 cubic meters per second, and it's decreasing from there. So we can see that the pool area is decreasing with lower discharge, while our riffle and run area is, well, riffles increase slightly and then decrease, while runs just decrease. So I took that data and I paired it with our aquatic insect abundances. So only looking at aquatic insects, and I've scaled the area of our samples by a meter squared. So I've done some calculations to figure out how many aquatic insects we have in a square meter of habitat. And so I've scaled that last graph by the aquatic insect abundances to give us the estimated reach-wide abundance of these aquatic insects for the entire Beasley study reach. So we can see we have the contribution of each of these habitat types, and then we have a line for total. So the total is just the sum of the other mean values for these habitats. So just looking at that total value, we can see that as our discharge is decreasing, we're going to start losing a lot of aquatic insects as their habitat becomes full. So some takeaways for question 3, how will aquatic insect abundances change under reduced flows? So we found that lowering flows reduced riffle and run area, but increased pool area. We also found that reducing the area of riffles and runs will likely reduce the total number of aquatic insects. So these communities that inhabit these riffles and runs, as their habitat becomes more like pools, we expect those communities to become pool-like communities that have less insects. And like I mentioned, the scope is a bit limited because we only have the Beasley study site here. Let's talk about some implications of our research. So we found that erosional and depositional habitats serve different functions in the Brady River. We found that riffles were important for aquatic insect production, and pools were important for contributing diversity to the system. We also found that if flows were to decline, we predict that aquatic insect production will also decline. And this is likely to negatively impact fish and other consumers in the terrestrial ecosystem that rely on these emerging aquatic insects as a food base. And we recommend that future management of the Brady River consider keeping flows high enough to maintain these riffle habitats, if at all possible, to make sure that the ecosystem is going to function how it should. So with that, I would like to acknowledge funding from the U.S. Forest Service that made this research possible. I'd also like to thank Oregon State University and the Department of Integrative Biology for supporting me during my time at grad school. I'd also like to thank collaborators, Dave Merritt at the U.S. Forest Service, Chris Holmquist Johnson at the USGS, and Joel Murray at the U.S. Forest Service. I'd also like to thank my committee members, Dave Lytle, Sally Hacker, Dana Warren, and Jeff Bethel for being here today and for giving me feedback throughout the process. I'd also like to thank my family for supporting me over the years and, you know, just being behind me every step of the way and, you know, making this possible. Shout out to my cats too for helping me complete my thesis, you know, sitting on my lap and playing on my keyboard, very helpful. I'd also like to thank past and present members of the Lytle Lab. So this research is really a team effort with every member of the lab, whether it's field work, lab work, you know, getting help with statistics or writing or feedback on my talk. You know, I was supported by members of the lab every step of the way, and I appreciate it. So with that, I will take any questions.
Hey, any questions? The committee members will usually save their kind of questions and comments for the defense that will follow this presentation. So.
Yeah. So you showed that you would expect that insect taxa would decrease in abundance as flows reduce. Do you think that non-insect taxa would decrease at similar rates, at a slower rate, or go up as pools increase? Like what would happen with non-insects?
That's a good question. I didn't look specifically at non-insects, but I would imagine a similar pattern might exist with non-insects just because pools were a little bit sparse in terms of the amount of invertebrates that we found. So yeah, I'd have to look at it specifically, but I imagine it would be a similar pattern there.
This is kind of related, but how do you evaluate the trade-off between increased like raw insect abundance in a habitat with mostly riffles versus what would, I assume based on your talk, be increased diversity in a mostly pool-based habitat? Like if flows reduced, do you expect that would increase the diversity of the system?
That's a good question. It's hard to know if fundamentally, like as these habitats are changing, our taxa that would normally be in a riffle, if they would no longer be able to persist there, like if that habitat is like qualitatively changing enough that they would no longer be there. Even if it meets our criteria for a riffle, it might be, you know, a low flow riffle. So I think overall, yeah, we would see diversity decrease because we're going to lose those riffle and run taxa in the system.
One of some question, it says, did you sample any prey of the insect larvae?
We sample any
prey of the insect larvae?
Yeah, so there are insects. So we don't specifically look at like the consumers of these insects like fish or birds or anything, but some of the insects do eat other insects. We did see some predatory insects like dragonfly larvae. There's some pretty gnarly insects called megaloptera that get really big and they eat tons of insects. Yeah, so we found insect predators, but not anything else.
It's another some question. As flows reduce, you will probably also get higher water temps and lower water quality. Do you have an idea of how much this might influence the changes you expect from habitat change?
Yeah, that's another important aspect. So we only really looked at the quantitative change in these habitats, but they are going to change qualitatively as well. While we don't have any data on that, I would expect that we're going to lose quite a bit of species as these water conditions change as well.
Any more questions? Yes. Okay.
Yeah, there is. The next question, it says, will it be worthwhile to factor in the biomass of adult terrestrial stages of indicators from each habitat type to estimate if lower abundance with low flows also means lower biomass available to terrestrial predators?
That's a really good question. Yeah, so another approach would be to collect the emerging aquatic insects and take their biomass to see, if there's less biomass coming from pools versus riffles and runs. Sort of tricky to do that and not something that we did, but yeah, I think that would be a good, we wanted to nail down this linkage.
Okay, sounds like there are no more questions online, so with that, let's give Justin another round of applause. And so now the committee will adjourn to discuss Justin's thesis, and also, if you haven't heard already, we're gonna have a party tonight over at my place. So please, as an attendee of this event, you're definitely welcome. So email me if you need any information on how to get there, and hope to see you all later. All right, take it away. Who are we meeting? You or?